I'm really, really pleased to introduce our speaker today. I should note that you can come to tea after over in the Center for Evolution and Medicine space, and we have the Evolutionary Biology Journal Club where we've read one of her papers, so maybe tea can transition into, you feel free to crash Journal Club and talk about the science a bit more, or science wherever you like all the time. That is what we do. So after this, please get tea and come to Journal Club. We'd be really happy to have you all at both. And so this is our first speaker for the spring series. So Montserrat Anguera did her bachelor's at UCSD in chemistry, and then did biochemistry working with folate metabolism at Cornell University, and then transitioned to studying X-Inactivation for her postdoc with Jeannie Lee, who's really just a leader in the field at Harvard, and then started her position as an assistant professor at UPenn in 2013, and we are both going up for tenure next year. So we feel the same pain, and we also love the same system, which is X-Inactivation, and I'm really thrilled to see the work that she does, which is really very different from what I do, and I hope you all will really enjoy the talk. Thank you very much for coming.
Thank you so much. Okay, perfect. So I want to thank Melissa for this opportunity to come here to ASU. This is actually my first time in Arizona. Usually I just visit the airport, but I'm really, really excited to see Tempe and to meet the students later on, and that we're going to be doing Journal Club. So it's actually should be really fun because I can answer the questions that you guys will have about the papers, and you could ask me about, you know, controls that are missing and stuff like that. So, in my lab, we study the epigenetics of the X chromosome, and we study a process called X-Inactivation. And the story that I'm going to tell you about today is the work that we've been doing to investigate how X-linked gene expression is regulated in female lymphocytes, so cells of the immune system. And what I'm going to be telling you about is that X-inactivation is maintained in a completely different way in female lymphocytes, contrary to all other female somatic cells. And what we found is that these cells have a very unique and dynamic mechanism to maintain the silencing of genes on the X chromosome. And what we are proposing in my lab is that this unusual form of maintaining silencing of the X chromosome is what may underlie sex differences that are observed in autoimmune disorders where the pathologies involve B cells and also T cells. So the outline for my talk today, first I'm going to tell you a little bit about X-inactivation, how the process is set up, and when it happens during development. And I'm also going to introduce you to Xist, which is a long non-coding RNA that plays a very important role in initiating and maintaining X-inactivation. Next, I'll share with you how X-inactivation is maintained in female lymphocytes. And then at the end, I'll show you some of the data that we've been collecting, looking at how the X chromosome is maintained during lupus, which is one of the female bias autoimmune disorders that we studied. So in my lab, we're interested in the biology of the X chromosome and how expression of genes on the X are regulated. So we know that there's differences in the numbers of sex chromosomes between males and females, where males just have one copy of an X and females have 2 copies of an X chromosome. And so the reason that we study this process, this regulation, is twofold. And so first of all, there's the regulation of expression of genes from the X chromosome is an absolutely fundamental biological process that's required for female development and female survival. And the second reason, and probably just as important, is that there's really amazing and interesting biology that takes place in order to regulate the expression of genes from the X chromosome. And so this involves epigenetics. And so the process of X-inactivation, I would say, is probably the hallmark best example of an epigenetic process, where you have this memory of silencing that gets transmitted with every round of cell division. And it involves all of the famous players of epigenetics. So you have DNA methylation, you have long non-coding RNAs, and you have marks of heterochromatin that all come together in order to regulate the expression. So why do we have X-inactivation? So the reason is for dosage compensation. And so if you look at between the sexes, the number of autosomes is balanced. So males will have 2 copies, and females will also have 2 copies. However, the sex chromosomes are not balanced between the sexes, where the females have 2 copies of an X chromosome and therefore are poised to possibly or potentially express twice as many genes from the X chromosome compared to males, which only have 1 copy of an X. And so what mammals have done during evolution is that they have developed this rather elegant and remarkable system where they will choose one of these X chromosomes at random for transcriptional silencing. And therefore, the dosage or the expression of genes between the sexes is balanced and equalized. And so that's why we have X-inactivation. It's for dosage compensation. So the transcriptional silencing of an X chromosome requires the long non-coding RNA Xist. And so I just want to take a moment to introduce you to this RNA. It is a 17 kb long transcript. It's never made into a protein, and so it functions as a long non-coding RNA. And it's exclusively expressed from one X chromosome. So it's expressed from the silent X chromosome, okay? And it functions in cis, so it is expressed and it silences the chromosome from where it's being expressed. And what's remarkable is that we can actually visualize this long non-coding RNA within the nucleus of a cell. And so here I have a picture of a nucleus. This is in blue DAPI staining for the DNA. And this is a cell that you would find in a female embryo pre-implantation, so before the embryo has implanted and started to go gastrulation. You will also see a cell that will look like this if you look at a female mouse embryonic stem cell. So in these types of cells, it's a very unusual situation where within the period of just a day and a half, you have 2 active X chromosomes. So both X's are firing simultaneously. You have an unequal dosage of genes being expressed, and you have low levels of expression of this long non-coding RNA Xist. And we can visualize this RNA. So there's about 4 or 5 copies of it being expressed from each X chromosome. And using fluorescence in situ hybridization, or FISH, we can detect the Xist RNA molecules within the nucleus of the cell. So these are these 2 pink pinpoints right here, and each one is an active X chromosome. So when the embryo implants and starts to undergo gastrulation, or if you take these female embryonic stem cells and differentiate them in culture, either of these active X chromosomes will initiate the process of X-inactivation. And so what happens is that you'll get upregulation of this long non-coding RNA from one of these X's, and the RNA will remain associated with the inactive X chromosome. And it forms this cytologically visible cloud, which we call an Xist RNA cloud. So this is the inactive X chromosome right here. And if you look at differentiated cells, this is what the pattern will look like. And all somatic cells, presumably, will look like this. So these are all cells that have undergone X-inactivation. So how does silencing take place? Well, one of the first processes in the formation of the inactive X chromosome is the expression of Xist. And after that event happens, Xist can recruit chromatin complexes that will come and deposit silent marks of chromatin all across the chromosome. And so the result is that you have this transformation of an active X chromosome into an epigenetically distinct inactive chromosome. So you can distinguish the active from the inactive X based on epigenetic modifications. And that's shown in this slide. So these are just a list of some of the features that the inactive X chromosome has. And these are features that you can actually see at the cytological level. So shown here on the right, this is the same nucleus going across these panels. And in green, this is a staining for Xist RNA, which coats the inactive X chromosome. So if you were to next do immunofluorescence for mark of heterochromatin, H3K27 trimethylation, you'll see that there's enrichment of this mark that perfectly co-localizes with the Xist RNA on the inactive X chromosome. The same is true for other marks of heterochromatin, such as macro-H2A. Also, these marks all shown in red right here. And so in somatic cells, all of these modifications function to keep the inactive X chromosome transcriptionally silent and in order to maintain dosage compensation. So I just want to take a moment and tell you about an unusual type of cell that doesn't have Xist RNA clouds. And these are human female pluripotent stem cells. And so these are a non-physiological type of cell, and they can irreversibly silence the Xist gene, and they lose Xist expression, and they also lose these modifications of heterochromatin on the inactive X chromosome. And so these are just a picture of what these cells look like if you use probes for Xist RNA FISH. So this is a population of Xist positive cells, and they have this robust Xist RNA cloud. And then in this population of cells, they have lost the ability to transcribe this gene, and they no longer have Xist coding the inactive X chromosome. And so down here is just a picture showing X paint, showing that these cells maintain 2 X chromosomes, and they lose the enrichment of marks of heterochromatin on one of the X chromosomes. And so as a result of this, gene expression changes in these cells from the X chromosome. And this is a RNA-seq experiment that we did to compare isogenic cell lines that express Xist and those that have silenced Xist. And you could see that across the autosomes, the transcriptional output is about the same. And the main difference happens when you look at the X chromosome, where Xist negative cells express a little bit more, but not twice as many of genes from the X chromosome. And so you need Xist in order to maintain X inactivation. And what happens, at least in the hematopoietic stem cell system, is that if you delete Xist, you get a female-specific, highly penetrant form of blood cancer that specifically targets the myeloid lineage. And in this experiment, this just shows the survival rate of animals that are homozygous deleted or heterozygous deleted for the Xist gene. And these are just pictures of, in this case, a wild-type spleen and female animals, either heterozygous or homozygous deleted for Xist, that exhibit really large splenomegaly. And what happens in these animals is that you get proliferation of the myeloid lineage at the expense of the other two blood lineages. So you need Xist in order to maintain X inactivation, in addition to initiating the process. So the function of X inactivation is to dosage compensate genes from the X chromosome between the sexes. And so what happens if this dosage isn't maintained? And so one of the really great examples of a disease state where this happens is autoimmunity. And there's about 80 different autoimmune disorders, and of those, 25 of them show a very strong female bias. And in this cartoon, I'm just showing you the risk for developing 2 autoimmune disorders which show a very high female bias, and those are Sjogren syndrome and also for lupus. So in these 2 autoimmune diseases, the number of X chromosomes that you have predisposes you to develop these autoimmune disorders. So for example, females that have 2 copies of an X, the prevalence is about 1 in 1,000 individuals that will develop lupus. Klinefelter syndrome, so these have 2 copies of an X chromosome and a Y chromosome. These individuals have higher risk to develop these autoimmune disorders compared to XY males. And similarly, individuals with 3 copies of an X chromosome are also at increased risk compared to XX females. And so conversely, if you look, it's almost impossible to find an example of an individual with Turner syndrome who has just 1 copy of an X chromosome that also presents with Sjogren syndrome or with lupus. And so one of the things that really caught my attention when I started thinking about this, when I started my lab, is that the X chromosome is unique among all of the chromosomes in that it has a really high enrichment for genes that are important or involved in immune functions. So for some reason, because of evolution, all of these immunity-related genes have concentrated on the X chromosome. And some of these are just shown here in yellow, and the ones that sort of stand out to me are TLR7 and 8, so these are toll-like receptors. And then also the CD40 ligand, which is responsible for T cells in order to activate B cells. FOXP3, which is an important regulator for Tregs, is also on the X chromosome. And so for some reason, mammals, like, we have done this. There's been a selection process that has happened. And so females having 2 X chromosomes, you can think about, are somewhat biased, perhaps, to have an enrichment of these immunity-related genes. But we have X inactivation that will dosage compensate. So in going through the literature, what's remarkable is that there's examples where the dosage of these autoimmunity or of these immunity-related genes from the X chromosome where they're altered. And so one of the examples is CD40 ligand. And so this was a study that came out in 2007 that compared lupus patients, male and female, and looked at the expression of the X-linked CD40 gene. And what they found was that women had about twice as much CD40 transcript compared to male lupus patients, where they were similar between healthy individuals and lupus controls. And so what the authors concluded in this study is that this increased dosage of CD40 ligand might be coming from the inactive X chromosome. They didn't look at this directly, but they just proposed this idea. And so when I started my lab, I started thinking about this. Could this in fact be coming from the inactive X chromosome? And we had all of the reagents in hand in order to start looking at this. Another example from the mouse literature, mouse models of lupus, where just to emphasize this idea of dosage of X-linked genes being important for playing a role in autoimmunity, is this mouse model. So this is the BXSB-YAA lupus mouse model. So in these mice, the males develop lupus. And it turns out that the reason for this is that these males have a translocation event that encompasses this region of the X chromosome that includes TLR7. So it's on the Y. So in effect, these animals have 2 copies of TLR7 compared to their healthy wild-type litter mates that would only have 1. So these animals will develop lupus, and it turns out that if you knock out one of the copies in this YAA model of TLR7, the animals are healthy and they never develop autoimmune phenotypes. And conversely, animals that are transgenic for TLR7, so if they have more than 2 copies, will develop lupus faster and with higher severity. So this just reemphasizes this idea of dosage of these genes to be very, very critical in autoimmune phenotypes. So in my lab, we think about this question a lot. Why is it that women are more prone to get lupus? And in order to start thinking about that, we wanted to ask the question of what does the inactive X chromosome look like in lupus individuals and also in healthy individuals. So in lymphocytes, is there any difference with the inactive X chromosome? And so when we started this project, a priori there's no reason why X-inactivation would be any different in lymphocytes compared to other cells in the body, right? They're all somatic cells. Once X-inactivation has taken place in the early embryo, it's getting maintained, presumably in the same way, in all cells of the body. That was our thinking. And so we started this experiment, and we just wanted to make sure that our reagents worked, and before we got our hands on the lupus control, or lupus patients. So we started by looking at Xist RNA FISH. So these are human female fibroblasts where we performed Xist RNA FISH. This is the probe in pink. And you could see that nearly all of the cells have a really robust cloud of Xist RNA that's present. So next, we started looking at different lymphocyte samples from healthy individuals. And these are just an example of human CD8 positive T cells, and we were shocked to see that there weren't any Xist clouds in these samples. And so we repeated this multiple times with different donors, and we would keep seeing that we wouldn't get an Xist RNA signal. Similarly, we looked at CD4 positive T cells, and in these cells we would see that there would be some clouds. It would be speckly, but nowhere near as robust as the human fibroblast cells. And in fact, this was conserved in mouse. So these are mouse bulk T cells, so CD8 and CD4 positive lymphocytes. And again, we saw like very faint clouds where 90 to 100% of the cells looked like this. And human B cells also lacked these Xist RNA clouds. And they, in fact, all of these lymphocytes tended to look like male cells, which don't express Xist RNA. And they don't do X-inactivation because they only have 1 copy of an X chromosome. And so this was the first example of a healthy primary cell line or primary cell type that didn't have Xist RNA on its inactive X chromosome. So something very unusual is taking place in these lymphocytes. So when we isolate peripheral lymphocytes or splenic cells, they're mature and they're naive, so they're not seeing antigen. And then what we can do in vitro is we can expose these cells to antigen and get them to re-enter the cell cycle. And so that's what we did here. So these are naive human T cells and mouse T cells, and we stained for Xist RNA, and in green is COT1, which detects nascent transcription of repetitive elements in the genome. And so we activated these cells in vitro using CD3 and CD28. And what we found, quite to our surprise, was that Xist RNA quickly returned to the inactive X chromosome in the cells. And so this is in human cells. And we would see that the Xist RNA was present in a COT1 negative hole region within the nuclear space, indicating that it was transcriptionally repressed. And so, again, this is another example where these lymphocytes are the first cells to have this dynamic type of maintenance of X inactivation, where this long non-coding RNA is somehow appearing here on the inactive X chromosome upon re-entry into the cell cycle. So the same thing is true in B cells. So these are experiments that we did using splenic B cells from mice, and we found that we can activate them in 3 different ways, and the Xist RNA signal just returns back to the inactive X chromosome. It's different kinetics compared to the T cells, and with different efficiencies depending on how you activate the cells in vitro. So what we observed is that with the activation, there's 4 different types of localization patterns for the long non-coding RNA Xist that we could see. And so I just want to introduce these different types of patterns because I'm going to refer to them during the later data in my talk. So the type 1 are what we call this robust Xist RNA cloud, where the majority of the signal is tightly localized in one area within the nucleus. Type 2 patterns are where there's a diffuse staining of Xist RNA, shown here in pink, that is within somewhat of a nuclear territory that would encompass the chromosome, the X chromosome. Type 3 is where there's pinpoints of Xist RNA that are found diffused all across the nucleus, and type 4 is where there's absolutely no signal whatsoever that we can detect by RNA FISH. So because naive cells are lacking a detectable signal within the nucleus of the cells, we thought, well, maybe there's differences in the transcription of the Xist gene that would account for the fact that these cells don't have anything, and then we see a signal again upon activation. And so what we did is we looked at unstimulated cells, and we looked at control cells of IMR90, so female fibroblast cells, and we were shocked to find that the levels of Xist transcription don't change at all in naive cells and activated cells. So there's constant and continuous transcription happening in human T cells. And in these human T cell experiments, we looked at the 5' end of the Xist RNA, because again, remember that the Xist RNA is really, really long. It's 17 kb. So there's about the same transcription at the 5' end as there is at the 3' end. So in B cells isolated from mice, the same thing. We see that naive cells transcribe Xist RNA at about the same levels as activated cells do. And so what this indicates is that the transcription of Xist is uncoupled from its localization upon activation. And so this is an idea that has been proposed in the field of X inactivation, that these 2 processes might be uncoupled. And these cells are the first physiological example to demonstrate that that is in fact the case. Okay, so what about the other modifications that are enriched on the inactive X chromosome? So we wanted to look and see whether the inactive X chromosome in lymphocytes, if it still had these other marks of heterochromatin that are normally detectable at the cytological level. So for these experiments, we performed sequential RNA FISH, followed by DNA FISH, followed by immunofluorescence. So in these images, this is the same nucleus across these 3 different staining conditions. And what we see is that the marks of heterochromatin are missing from the X chromosome in the naive state. And these marks return to the inactive X chromosome upon activation. So these results here are for human T cells. We see the same thing in B cells from mice and also from humans. So some of these marks, in particular H3K27 trimethylation and H2A ubiquitin, come back to the inactive X chromosome about 80% of the time. What's curious is that there's other marks of heterochromatin that don't really associate with the inactive X chromosome in activated cells. And so what we conclude from these experiments is that the inactive X chromosome doesn't seem to be as tightly silenced as it is in somatic cells. And so this is just sort of at a broad, zoomed out level. So next we wanted to test this idea of whether there might in fact be genes from the X chromosome that are escaping silencing in lymphocytes. So for these experiments, again, we're using healthy lymphocytes. We just picked 2 X-linked genes that are important for immunity and autoimmunity. So CD40 ligand and CXCR3. And what we did is RNA FISH, where we used a probe specific to these X-linked genes, together with a probe for the long non-coding RNA Xist, which would identify the inactive X chromosome in the activated cells. And then we counted the number of pinpoints, so nascent transcription from the X chromosome. And so if there was 1 pinpoint, this would reflect monoallelic expression of these X-linked genes. And the presence of 2 pinpoints would reflect biallelic expression, so expression from the inactive X chromosome in addition to the active X chromosome. And what we found was that for both of these genes, the majority of the cells were monoallelic, as we would expect. However, there was a small population of cells that had biallelic expression of both CD40 ligand and CXCR3. And these biallelic expressing cells, one of the pinpoints would overlap with the Xist RNA cloud, just like this. And so what this indicates is that the silencing of the X chromosome is in fact a little different and more relaxed in these lymphocytes. So what about the rest of the X chromosome? So we wanted to test the hypothesis that if there's partial reactivation happening from the inactive X chromosome in lymphocytes, if we were to compare healthy female individuals with 2 X chromosomes with healthy male individuals with just 1 X, and that being an active X chromosome, if we look at increased expression of X-linked genes between the sexes, presumably that might be coming from the inactive X chromosome. Does that make sense? So it's indirect by comparing the two sexes. So we went through the GEO databases and we found a study where they had looked at naive mature peripheral B cells in human male and female individuals. And we just compared male to female. And the results are shown here. So there were 3 regions on the X chromosome that really distinguished the 2 sexes. And the most statistically significant region between males and females is the Q13. And this region encompasses about 25 genes. And Xist is also present in that region. And so that's sort of our positive control, right? So we wouldn't expect to see that in males, but we would see robust expression of it in females. And so in fact, that's what we found is that Xist is predominantly overexpressed. And then curiously, we found that there were 2 immunity-related X-linked genes also within this region whose expression was higher in females compared to males. And so what this suggests is that there seems to be more expression happening from the inactive X chromosome in these lymphocytes who are just healthy as compared to other somatic cell types. So we wanted to test that. Okay, so if maybe this is just something that happens when you compare sex differences, irrespective of a lymphocyte population. So we repeated the experiment and we looked at fetal lung fibroblast samples. And so compared male to female, and we didn't have as many samples as we would have liked for the B cells. So 1 male but 3 females. And we found that the same region, the Q13 arm of the X chromosome, is the region that most distinguished the males from the females. And again, we see Xist is highly enriched. However, what's significant here is that all of the genes that pop out in this analysis, none of them have anything to do with immunity. And these are all genes that are known to escape X inactivation. So they're known to be expressed at twice the dosage in females compared to males. And so what this indicates is that our findings that there's something different about the regulation of the inactive X chromosome in lymphocyte cells that's taking place compared to other somatic cells. So one of the questions that we would get asked a lot when we started working on this early on is when does Xist RNA first disappear from the inactive X chromosome during lymphocyte development? So we don't see it in mature B cells and T cells, but how far back does that go? So what we did in order to answer this question is we isolated subsets in the bone marrow, which is where the progenitor cells are for all the lymphocytes. And using flow, we isolated all of these different subsets that give rise eventually to mature follicular B cells. And what we found was that the progenitor populations in the bone marrow all had Xist RNA clouds, and these are representative nuclei right here. And then there was something very important that happened during the transition from the common lymphoid progenitors to the pro-B cells. And what happens is that this Xist RNA cloud completely disappeared. In about 100% of the cells, they completely lacked Xist RNA localizing to the inactive X chromosome. And then Xist stayed off or undetectable in the lineages up until about the plasma cell stage. And that's when we start to see detectable signals again. So we asked whether Xist RNA transcription might be changing in these cells. And it turns out that there's a continuous transcription of Xist RNA during B cell development. But there's just something preventing its localization that's happening in the pro-B cell stage. So next we looked at marks of heterochromatin during B cell development. And what we see is that in the pro-B cell stage, there's still foci for the two marks of heterochromatin, H3K27 and H2A ubiquitin, that are present in the cells, even though there's no Xist signal detectable in these cells whatsoever. And so as these cell populations differentiated to a mature B cell stage, the marks of heterochromatin disappear. So we start to lose these foci. And what's curious and what we still don't understand is like why these cells have evolved this relaxation. Why are they getting primed for different regions to become reactivated and losing these marks of silencing? So in the next part of my talk, I want to tell you a little bit about mechanism. So we were really interested in this idea of how Xist RNA gets to the place where it needs to go to, and what factors are regulating this process and the dynamics of the return of the RNA to the inactive X chromosome. So we did time course experiments where we followed the localization of the RNA to the inactive X chromosome in splenic B cells. And what we found is that the RNA comes back to the inactive X in two phases. So in the first phase, what we see is that there's RNA signals that are spread throughout the nucleus in a very diffuse pattern, and we call this phase 1. And then the second phase, which happens at about 16 hours, is when the RNA pinpoints start to localize on the inactive X chromosome to start forming these kinds of nuclei. And so that's kind of shown in this diagram. So at the top, this is the result that we see by Xist RNA FISH, and this is our model for what we think is happening. So we know that Xist is being continuously transcribed in this state, but we're not detecting it by RNA FISH. And so what we're thinking is that perhaps the RNA is very relaxed and it's disordered, and just one molecule perhaps we're not able to detect it using RNA FISH. However, there might be a protein that is binding to Xist RNA upon activation, where you can bring together perhaps 2 or 3 transcripts, which then we can detect using Xist RNA. And these are why we see pinpoints that are distributed all across the nucleus. So this is what we see at phase 1. And then at phase 2, these proteins, perhaps interacting with each other, together with Xist RNA, are localizing it onto the inactive X chromosome. And this is happening between 16 and 24 hours, and it looks like this in the fish. What we see is that the marks of heterochromatin return to the inactive X at the same time that Xist is coming back and localizing within this nuclear territory. So in these experiments, we performed time course experiments after activation, and we quantified the number of cells that co-localize with Xist. That's shown right here in blue. And we can see the co-localization increases with time. And then these are just representative nuclei where you can see a heterochromatic mark that overlays with the Xist RNA signal. So next we asked whether Xist is continuously transcribed in naive and activated splenocytes, and whether the stability of Xist RNA transcripts within lymphocytes influences its localization at the inactive X chromosome. So for these experiments, we use actinomycin, which is an inhibitor of RNA polymerase, and we see that once we give the cells actinomycin, RNA polymerase gets inhibited, and Xist RNA transcripts are rapidly degraded. So what this indicates is that there's continuous and very fast transcription happening at this gene in naive cells. And we calculated the half-life to be about just under 2 hours, which is rather significant because the half-life of Xist RNA in embryonic stem cells and other somatic cells like fibroblasts is about 6 or 7 hours. So these lymphocytes are turning over Xist really, really quickly. So we repeated the experiment in activated splenic B cells, and again we see the half-life is pretty similar. And at 24 hours following treatment, we looked to see where Xist was within the cell, and what we found, as we would expect, there was no Xist RNA signal on the inactive X chromosome, indicating that you need continuous transcription of the RNA in order to keep it localized on the inactive X in activated splenocytes. So what is bringing Xist back to this region, this X chromosome? For these experiments, we took a candidate approach and we went through the literature and looked at known Xist RNA binding proteins that play a role in the initiation or the maintenance of X inactivation. And so we had 2 candidates, and what I'm going to tell you about in detail today is one of them, and that's YY1. And so this is a transcription factor. It's called YY1, and the reason it has this name is that sometimes it functions as an activator, sometimes it functions as a repressor protein. And so it has a number of roles in nuclear architecture and organization, and in B cells, it's very important in class switch recombination. So we first asked whether YY1 might be required during T cell activation in order to bring Xist back to the inactive X chromosome. So for these experiments, we used human naive T cells, and we transfected in siRNAs that were specific to YY1 and a scrambled control. Then we stimulated the cells and harvested them at day 2 and 3 post-activation, and then looked to see whether the patterns of Xist RNA were different when the YY1 protein was reduced. And so these are the results for a scramble, and so we could see that there's the presence of robust Xist RNA clouds in activated T cells, and this is a zoom in of one of these nuclei. So when we knocked down YY1 using siRNAs, what we found was that the majority of the cells did not have robust Xist signal on the inactive X, and instead we saw that the signal had dispersed pinpoints all across the nucleus. And that's quantified here for days 3 and 4, so days 2 and 3 post-activation. So you need YY1 in order to get Xist RNA localized to the inactive X chromosome during T cell activation. Next, we wanted to repeat the experiment in using B cells. So we worked together with Mike Atchison, who's one of the people who discovered the YY1 transcription factor, and he has a really elegant system where using YY1 flox animals, we can introduce the TAC-CRE protein into the naive splenocytes. And this TAC-CRE will then perform the recombinase reaction to delete the YY1 in ex vivo. And then after that, we stimulate the cell. So all of our activated splenocyte population is lacking YY1 protein. So we collected the cells at this stage, and then we performed Xist RNA FISH to see if deletion of YY1 would affect the localization of Xist to the inactive X chromosome. So these are the wild type cells right here, and these are the YY1 deleted cells. And what we see is that upon YY1 deletion, we see this dispersed pattern of Xist RNA staining across the nucleus. And we see that the majority of the cells are missing the robust Xist RNA clouds. And those results are quantified right here. So next we wondered, well, if we're if we're deleting YY1 and we're preventing Xist RNA localization to the inactive X chromosome, what's happening to these marks of heterochromatin that typically co-localize with Xist RNA at the inactive X chromosome? So we repeated the ex vivo deletion of YY1 using the splenocytes TAC-CRE system. And these are the wild type results right here, where we can see co-localization of Xist RNA with H2A ubiquitin or H3K27 trimethylation. When we delete YY1 in splenocytes, we lose the foci. There's some cells that we could detect a foci, but these are ones that still had a little bit of an Xist cloud. And so the results are summarized right here. So you need YY1 in order to bring Xist back to the inactive X chromosome and in order to keep these heterochromatic marks enriched at the inactive X chromosome. So next we wondered, well, are there any consequences to gene expression from the inactive X chromosome when we delete YY1 in activated B cells? And for these experiments, we wanted to look specifically at the inactive X chromosome. So we used the same approach where we compared different sexes. So if we compared YY1 deleted male samples compared to female samples and then looked specifically at the X chromosome, if we saw genes that were higher or lower expressed in the female animals, this would be reflective of changes happening at the inactive X chromosome. So we performed RNA-seq experiments in YY1 deleted male and female animals and also wild type male and female animals. And these are results looking at the differentially expressed genes in YY1 null B cells. And what we found was that there were 68 female-specific genes whose expression was changed when we deleted YY1. There were 11 genes that, these are just on the X chromosome, 11 genes that were shared between males and female mice, and there was one male-specific gene on the X chromosome that was unique to males. So these 68 genes break down into this is a heat map clustering showing the differentially expressed genes. 20 of them were upregulated and 48 of them were downregulated. So of this 20 that were upregulated on the X chromosome, the top 15 are shown here. And what's really exciting for us is that we continue to see immunity-related genes on this list, including CXCR3. There's a complement factor protein right here. So we're now starting to look and see, like, what is it about these genes that might predispose them to get reactivated when Xist RNA isn't getting back to the inactive X chromosome. Okay. And so YY1 is a really interesting and unusual protein because it functions as an activator, functions as a repressor. And so there's been a ton of biochemical studies that have dissected out all of these different activities for this transcription factor. And what we know is that these different activities are located on different domains of the protein. So this is the YY1 protein, and here are just representation of the different domains that it has. And so at the C-terminal end, it has 4 zinc fingers, and this is how it binds to DNA regions. There's a transactivation domain right here at the N-terminal end. And there's a couple of studies that have suggested that either one of these 2 domains, or perhaps both, can bind to RNA directly. Through the HDAC domain, it recruits histone acetyltransferases and deacetylases. And the repo domain is important for recruiting actually the PRC2 complex, which is responsible for putting down the H3K27 trimethylation marks for heterochromatin. So we wanted to ask the question, which of these domains on YY1 is actually necessary for bringing Xist RNA back to the inactive X chromosome? So what we did is a series of rescue experiments where we deleted YY1 and then we added back truncations of the YY1 protein that contained different regions of these domains shown here. And so what we found was that the zinc finger domain was the only region that was necessary in order to bring Xist RNA back to the inactive X chromosome. And that's shown right here. So adding back the zinc finger, we see clouds, and it's quantified right here. So deleting all of these other regions and then bringing them back was not sufficient to do anything. So there's something about this zinc finger domain that's important for recruiting Xist back to the inactive X chromosome during B cell activation. And so right now we're starting to think about like what proteins perhaps interacting with YY1 are binding to Xist RNA in order to bring back this RNA protein complex back to the inactive X chromosome. And so our model for this process kind of looks like this, where Xist is continuously transcribed in naive B cells, but there aren't enough of the binding proteins present in order to cluster the transcripts together and keep it tethered to the inactive X chromosome. And so this is why the RNA will start to diffuse away and be found all over the nucleus. So upon stimulation, there's the translation of proteins that can bind to the Xist RNA transcript and cluster together. These are just hypothetical proteins that I have in blue in this cartoon. And we think that these proteins are different than the YY1 protein, and that the YY1 protein is functioning, and it's important for this second phase of recruiting Xist RNA back to the inactive X chromosome. And YY1 might be interacting directly with the Xist RNA transcript, or perhaps YY1 is interacting with Xist binding proteins in order to bring the RNA back to the inactive X chromosome and then keep it tethered there. And that visually would look like this in Xist RNA FISH. And all of this is happening within 24 hours. Okay, so in the last 5 minutes of my talk, I want to tell you about another story that we're working on in the lab, and that is to look at lupus patients and also lupus mouse models, and to see what is happening with the inactive X chromosome, if there might be any differences, silencing differences, epigenetic differences on the inactive X chromosome during autoimmune disease. And so the first sort of hints that we got that suggests that there might be something different happening came from immortalized B cell lines that we got from the Children's Hospital at Philadelphia. And we had a small sample size at that point. But what we saw is when we quantified the patterns of Xist RNA localization, in lupus patients, they seem to have fewer cells where Xist RNA was tightly localized on the inactive X chromosome. And that's quantified below right here. And there was a lot more of nuclei that looked like this, where the Xist RNA signal was diffuse all over the nucleus. And there was also a lot of cells that completely lacked a detectable Xist signal across the nucleus. And so this was some of the first stuff that we published in our original paper, and we only had like 5 cell lines. And so this was really the data that prompted us to write an IRB, which is a huge pain. I don't know if anyone's had to do this, take this upon themselves. But this was the motivation for me to really like get on it and write this IRB so that we could look at primary samples from patients and look to see what was happening with the inactive X chromosome. And so what I'm showing you now are just like very, very preliminary data that we're starting to collect. And so these are the results from pediatric patients from the Children's Hospital of Philadelphia. And here we're looking at T cells. And what we're starting to see is that the patient cells have mislocalized Xist RNA patterns compared to their healthy age-matched controls. And so here, this is a picture of activated CD4 and CD8 T cells from a lupus patient, and you can see that the Xist signals are sort of more diffuse and as compared to the healthy age-matched controls, where they have a lot more nuclei that have very robust Xist localization on their inactive X chromosome. So what we're in the process of doing right now is looking at T cells and also B cells to see if this holds up. What are the percentages of co-localization with different marks of heterochromatin? Do we see biallelic expression of X-linked genes? All of this stuff is ongoing. So we're just right now collecting samples, and stay tuned for that. The last thing I want to share with you is some of the studies that we've been doing using a very interesting classical mouse model of lupus, and this is the NZB-WF1 mice. So these are mice where you mate a New Zealand black, and these animals have a black coat, to these New Zealand white animals, and it's I think it's kind of cool. It's kind of crazy. The F1 generation develop a female bias to develop autoimmune-like symptoms. So this happens in the F1s, where these animals in the serum over time will develop circulating double-stranded DNA autoantibodies, Their kidneys will start to break down, so you'll see proteinuria in the urine, and you'll get the formation of immune complexes, which will start depositing in the kidneys, and these animals will eventually die of glomerulonephritis. And so this has a lot of similarities with lupus patients. And so we wondered, okay, well, if this has a really strong female bias, maybe there's something going on with the inactive X chromosome in this mouse model for lupus. And so we're just, again, this is all unpublished and we're just in the early phases of this study. These are the results from animals with late-stage disease. So these are wild-type animals right here for Xist RNA fish, and these are the disease animals. And you'll notice that similar to the lupus patients, their Xist clouds are not as robust, and there seems to be less localization, concentrating in one nuclear area. So we've been quantifying the different localization patterns for the mice, pre-disease, early-stage disease, and late-stage disease. And if you look at in bright yellow, these are the type 1 clouds, the robust Xist RNA clouds. What we see is that the numbers of the percentages start out being about 30 to 40% for the wild type and the F1 animals. Then mice with early stage disease, the levels start to kind of drop a little bit, but it's not statistically significant. And then animals at 9 to 11 months, so with late stage disease, the F1 animals have much lower percentages of robust Xist clouds. So the way that we're thinking about this is that it's an Xist RNA localization thing that's happening in lupus that is predisposing these regions of the X chromosome to become reactivated. Not the whole X chromosome, but just different parts of it. Okay, so just to summarize my talk today, I told you about how lymphocytes have this very unusual and dynamic way to maintain X inactivation, where in the quiescent state, they don't have Xist RNA and marks of heterochromatin that are localized or enriched on the inactive X. And upon activation, these marks come back. I told you about how this process is dynamic and it changes during B cell development and lineage commitment. And with regards to mechanism, we found that YY1 is one Xist RNA binding protein that's important for recruiting and tethering the Xist RNA to the inactive X chromosome. And finally, using mouse models and patient samples, we see that there's differences with the degree and the amount of colocalization or localization of Xist RNA to the inactive X chromosome. So with that, I just want to take a moment and thank all the people who do the work in the lab, who are doing all of these fish experiments and all of the counting in the microscope. This is my lab right here. Past and former member of this project was started by Janla, who is a wonderful postdoc in the lab. At Penn, there's a really great immunology community, and I would not have been able to do these studies without all of their help. So some of these individuals are listed here. So with that, I'd love to take questions. Thank you.
We'll use the microphone just for posterity.
So very interesting and provocative.
Thank you.
Are there other tissue types besides the lymphoid tissues that show this this pattern? And is there any speculation as to why or what's going on with that?
Yeah, that's a really great question. So, we've been looking at other subsets with the immune system. Neutrophils do a similar but different thing. So neutrophils are interesting because they're terminally differentiated, but they also exist in quiescence. And so when they get activated, they will re-enter the cell cycle, but then they will only exist, I guess. They'll only be alive for about 24 hours. And then they, explode, right, form these nets and stuff. So what we found is that, so the resting neutrophils don't have any Xist RNA. They're expressing Xist, but we don't have any detectable localization on the inactive X chromosome. There's no enrichment of the marks of heterochromatin. Upon activation, we see like 1 or 2 cells that will start to have diffuse Xist RNA signals across the nucleus. But that's it. We never see robust clouds. And then they just, yeah, kind of start exploding.
So do you think it's a hallmark of rapidly dividing cells, or is it a hallmark of cells that have the capacity to do since you were looking at it in pluripotent stem cells as well?
Yeah,
and
yeah.
Just out of curiosity, if you looked at, for instance, liver or any other cells that actually have tremendous capacity for
regeneration.
Yeah.
Yeah, so that's a really good question. So I had we have I had a rotation student who now thankfully joined the lab, and this was her rotation project. Like, look at other cells outside of the immune system that are quiescent. What happens, right? Or what happens, like all the classic experiments, classical ones where you do serum deprivation, right? Then you get the cells to exit the cell cycle, go into G0. So the short answer is that there seems to be something about the state of quiescence that sometimes dictates this, even outside of the immune system. But that's not always the case. So for example, it depends on what cell you are. If you do serum starvation with MEFs, there's absolutely no change with Xist. It'll stay localized to the inactive X chromosome just fine. And you bring serum back. It's like nothing. We've also looked in the satellite muscle system. So that's probably, like, I would say, the best defined in the terms of quiescent adult stem cells, right? The satellite cells don't have Xist RNA. Then when you differentiate them into myoblasts, that's sort of the first progenitor stage after that, they have the Xist clouds. So identical to what we see in the T cells and the B cells. And so now we're thinking about, like, intestinal stem cells. What are they doing? The HSC system too. It has the long-term and short-term cycling ones. So we think it's a cell cycle thing that's happening. Yeah, thanks. Any other questions?
I have so many. I'll start with two.
Okay.
Or one. How difficult would it be to have a mouse model of Turner syndrome to use to compare? Because you did XX versus XY, but to add in the X naught to it, is it just?
Yeah, people have been trying to do Turner mouse models.
They just don't work.
And
it
doesn't work, even though, like, humans, like, we can do it. Yeah. And it's mosaic in humans, so the degree of mosaicism for Turners.
Well, so I was wondering, because their Y chromosomes have lost so many more genes, if maybe you have, and then they have more inactivation than we have.
Right.
With less escape. So we're more, we have 30,
we're more leaky.
Yeah, we're more leaky, like 33% or something. So then maybe Turner syndrome would have a more severe phenotype. And anyway, okay. And then maybe I'll use this as a time to break and go to tea and journal club. So with that, let's thank our speaker again.
Thank you.